So in the last video, we talked about the receptive field of neurons at various parts of the visual pathway. Like rods have a receptive field for just a point of light hitting where the rod is on the retina, and ganglion cells have center surround receptive fields, basically just covering a patch of the retina made up of a whole bunch of rods that the ganglion cell is connected to. That sends its signal through the LGN of the thalamus, stopping at a neuron that has the same center surround receptive field as the ganglion cell it's getting its signal from. And finally, we came to V1, primary visual cortex at the back of the brain, where we found simple cells, which are just neurons that fire for edges, for little lines at a particular orientation at a particular part of the visual field. But within V1, there are some other cells that actually look for slightly more complicated things, like a moving edge or a combination of edges, things like that. For example, Hubel and Wiesel, those Nobel Prize winners who mapped out all this stuff, they also found some neurons that they called complex cells. Complex cells respond only to moving edges or moving lines, and generally only for motion in a particular direction, although some are not direction sensitive, they would go either direction. So like in this bottom image here on the right, if a line is tilted just the right angle and moving down and to the right, it's going to make this one specific neuron back in V1, a complex cell back in V1, will go off. It'll fire a lot if that is happening in front of you. So the complex cell goes pew, 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 pew if we're measuring it with an electrode, say. But if you take an edge or a line that's at that exact same angle in the exact same part of the visual field, but moving in the opposite direction, this neuron may not fire. We'd say it's a direction selective complex cell. Here's another example. The little lines on the right here, those are the action potentials. So you're just seeing kind of the action potentials, the spikes of the neuron going off. The horizontal line, like moving downward, that does nothing. Tilted a little bit, might cause a little activity, kind of getting warmer. But then when you reach the correct angle, the one this neuron is programmed for, the one it looks for, now downward movement sets off this complex cell like crazy. But if you tilt it even further, so now it's like a vertical line moving sideways, not much activity. And likewise, if the movement's in the wrong direction, the cell won't fire. So that's the idea of complex cells. Basically, just think of it as lines. They're just like simple cells, but for moving lines. Beyond simple cells and complex cells in V1, there are also neurons called end-stopped cells, which respond specifically to corners, or to corners moving in a particular direction. So in this image, think of this like a white piece of paper on a blue background, and you're moving the white piece of paper upward. Like in the top part of the image, a piece of paper is too far to the right. So when it's moved upward, this particular neuron doesn't care that much. In the very bottom image, the piece of paper was already too far to the left. So when it starts moving upward, this particular end-stopped cell doesn't really care. But if the piece of paper is halfway between, if that angle, that corner is in just the right place where this end-stopped cell is watching that part of the visual field, in other words, if the corner is in its receptive field, then when the paper starts moving upward, this neuron would go pew, 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 and fire a whole bunch, saying, I've detected a moving corner, not just a moving edge. So think of this as just slightly more complicated than a complex cell. So now we've gone from simple cells that detect lines to neurons that look for particular combinations of simple cells firing. Like when we talk about detecting a moving line, we're really just saying we need a neuron that detects when a series of other neurons all go off in a row. So for a horizontal line moving downward, it needs to detect a horizontal line at the top, then slightly after that, a horizontal line a little bit lower in the visual field, then a horizontal line just a little bit lower, and so on. If those simple cells fire in the right order, right, then this complex cell, the one I've drawn in purple here, will go off because those simple cells will only fire in that order if there's actually a horizontal line moving downward. So voila, we have a moving line detector. Obviously, we'd need to build a little neural circuit between these simple cells and the complex cell to ensure that it, only turns on if they fire in the right order. But we saw a basic version of that back when we learned about adaptation, so that's not that hard to wire up. The big point here is just that our brain's ability to recognize anything, like trees and houses and dogs and Salvador Dali paintings, it relies on registering simple features with early neurons, kind of early in the process, and then registering more complicated combos of those simple features in the form of complicated combos of activity in the earlier neurons. So we build complex things out of simple pieces, all of it instantiated in neurons that fire only in one specific situation. that have a receptive field where they're sort of indirectly watching that one part of the visual field for that one specific thing via all those simpler neurons that they're directly watching. So let's just quickly reiterate the receptive field of neurons we've met so far on our path in sort of out of the eye and into the brain. And then we'll see some footage in a moment from those original Hubel and Wiesel experiments that discovered these receptive fields. So coming out of, I got those backwards there, sorry. Coming out of the optic nerve of the eyeball, so at the top here, are the axons of ganglion cells. Those axons will only be active. Those ganglion cells will only be firing if some light happened to fall on the patch of retina that ganglion cell is watching, specifically in the correct part of its center surround layout, made up of a bunch of rods. That axon, that ganglion axon, synapses onto a neuron, connects onto a neuron in the LGN, which fires if the ganglion cell fired, so that LGN has the same receptive field as the ganglion cell itself. Then we're into the cortex, right? We're into V1, and we've got simple cells. Again, the layout of the wiring is basically set up to have an excitatory area all in a row and little inhibitory areas on each side of it in a parallel row. So this cell will only go off. It's very specific. It only goes off for a line, an edge, a bar, showing up in just the right place at just the right angle. Then we've got complex cortical cells, or just complex cells, if you want, which respond best to movement of a correctly oriented bar across that part of the visual field. And finally, those end-stopped cells, which, again, generally only fire for a specific corner or angle, moving in a particular direction. So, okay, I've got a few video clips here from the original experiments mapping out receptive fields. In this first video, you'll see them recording from an LGN neuron. So they're in the thalamus, they've done surgery, gone into the thalamus, and they're recording on an individual neuron, trying to figure out what makes that neuron go off. They're using an electrode, so what you're going to hear, the sounds you're going to hear is static. That static, kind of static sounding thing, is the action potentials of that one neuron. So it's just a recording of the action potentials, the firing. So if you hear a lot of static, that's a lot of firing. If you hear silence, that's no firing. So you can hear it fire many times per second when it's getting stimulated in the right way, whereas otherwise it might fire just a little bit, kind of background, baseline, or partial stimulation. Just a side note, the audio in these, they're really old clips, so the audio is out of sync a tiny bit. So you'll actually hear the staticky sound of neural activity just a little after the light turns on in the correct place, after the stimulation hits the receptive field. But in reality, they were totally synced up. It's just an old film. So here we go. I'm going to play. You'll see they've already drawn a little circle in the middle with a marker on the screen mapping out this neuron's receptive field. Again, the sound is a little delayed behind the light. So this one neuron they're recording from seems to fire most heavily when the light falls directly on the center bit, and less if it's hitting that outer surround. So if the light falls specifically just on the center, we get maximum kind of static sound, maximum firing. When they try that big diffuse light, it's not as much activity. This doesn't work very well because it's not hitting the center where it's really excitatory. By the way, to make these shapes of light, they were literally just using an old projector and sort of manually waving different shapes of light around on the screen in front of the animal that's just staring forward without moving its eyes. And what you're hearing is literally just a single neuron, a single wire. They're just measuring an axon of one of those LGN neurons. So when the light goes through that center part, it does cause some excitation, but not as much as if the light was selective to there, right? Because it's also hitting some of that inhibitory surround. Let's see another example here. Yeah, we'll go to this one. All right. So here they've moved an electrode, so it's hooked up to a random neuron at the back of the occipital lobe back there in V1. So they happen to have grabbed a simple cell, but the only way they know that, the only way we know it, is to shine various types of light on the screen in front of the animal and see what makes this neuron fire. So here they've kind of already found the area where it seems a bar of light seems to make this neuron fire. And you'll see them actually mark on the screen the area of the visual field that this particular neuron seems to be watching for that line pattern, its receptive field. Not much activity off on the side because it's the inhibitory part next to it, right? It only goes off if you fall right on that excitatory part. So they're sort of testing around to find the boundary, and you'll see him actually mark the inhibitory areas around it. Eventually, they've kind of mapped out exactly where the light needs to be and the shape of the light to make this neuron fire at its maximum. So they're mapping out its receptive field. Now they're going to try the bar at different angles. Notice the line has to be at the correct angle. Right? You don't get much activity unless it's at the correct angle. There it worked. Again, the sound is a little delayed because it's old film. You could also just try like a big light everywhere and see what the neuron does. Now they'll do a big diffuse light, and you'll notice the neuron's probably not going to care much, right? Because it doesn't have a line, it doesn't have an edge, it doesn't have a place of contrast all in a row when it's just this big diffuse light hitting everywhere. So even though the right place was lit up, it wasn't like a line, it wasn't contrast. But if there's an edge there, that's what it cares about. That's why I say they're sort of edge detectors. When I say line detectors, really what I mean is any sort of edge or place of contrast all in a row, which doesn't happen with the big diffuse light, but does happen there when they got an edge. So you kind of get the idea. All right. Let's do, this video will show a complex cell. So this is still in V1, a neuron back in V1, just happens to be a complex cell, meaning it fires only for a moving edge. So let's get this one started. You'll see them mapping out kind of how big of a patch of the visual field this neuron is sensitive to. They've already obviously been testing around different angles and places of putting the line. But now they're just sort of mapping out how big of an area that moving line that this cell cares about or watches for, its receptive field. And remember I mentioned some complex cells are direction sensitive. They only fire if the line is moving in one direction, but others fire if the line is moving in either direction. They're not all direction sensitive. So I'm mapping it out, trying to find where this one first starts carrying. And now they can get it down to the area it's watching. Like I said, some of them are not direction sensitive. They'll fire for either direction of movement as long as the line is angled correctly and in the correct part of the visual field. So we draw this kind of an arrow tip on both sides saying this is whether it's left or right movement works for this one. But if the angle is all wrong, not much activity. Right, not nearly as much or close to none. So that's a complex cell. I can show you one more. Finally, this clip will just show them recording from a different complex cell in the back of the brain and seeing what that one fires for. Here they found a complex cell that is direction sensitive. So in this case, the line has to be moving in a particular direction. This one seems to maximally fire only for that one direction of movement at that angle in that part of the visual field. Again, it's a little hard to tell with the sound delay here, but so yeah, that's them testing a complex cell. So you kind of get the idea. They just did a bunch of experiments like this, like a whole ton of these. But remember, each time they actually did surgery into a cat's brain, and they're moving an electrode from one neuron to another neuron and trying to then, while they're recording that one neuron, and all you get from that neuron is whether it fires or not, right? You get the little spikes, the little pew, 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 or as you can hear here, right, staticky sound. That is all that they have while they just do some things out there in the environment and see what is it that this one neuron cares about? What makes this neuron fire? That's why I say they're mapping out the receptive field of these neurons that they're recording in the brain. And we're already on to things that are quite a bit more complex than just a dot of light that turns on a rod or cone, and up to where we can detect things like a moving line in a particular part of the visual field at a particular angle. And so combinations of that might lead to us being able to track, you know, a moving set of other features, which might be like a tiger moving in front of us or something moving towards us or other detection of motion. That's kind of the idea, as we map out what these neurons do and how they're hooked up to more neurons that look for more complicated combinations of those simpler things. That's how we get a feel for how the brain, you know, recognizes complex stuff. So from V1, that area in the back of the brain we've been talking about, where the simple cells, complex cells, and interstop cells are, from there, visual input goes to other parts of the brain. So initially it's going to go to a bunch of other areas in the occipital lobe. There are like around 30 or so, depending on how you define it. There are around 30 or so different areas that are visually selective. Not all of them in the occipital lobe, some sort of at the border, occipito-temporal or occipito-parietal, but like there's a bunch of specialized modules, we could sort of say, within the occipital lobe for processing visual information. So it goes from V1 to areas with fancy names like V2, V3, V4, and then some other areas like MT is for motion perception, for example, and things like that. But from the occipital lobe, those purely visual areas, the info also goes to whole other lobes of the brain. For example, some of it will go sort of down and forward to the temporal lobe. Some will go up and forward to the parietal lobe, basically sharing the visual information with other parts of the brain so we can use visual information to do tasks. And you may remember the side of the brain, the temporal lobe on the side of the brain, that's important for recognition, for categorizing things, right? In addition to hearing and language. And then up here, the parietal lobe is really important for spatial stuff, right? So it might be using vision to do spatial tasks. So the info gets shared so we can actually do things with that information, like recognize stuff, make decisions, eventually goes to the frontal lobe, right? So we can plan or connect it with, you know, information across time, stuff like that. Now, as we go further into the brain, we still find neurons with very specific receptive fields, now looking for even more complex combos of stuff. So here on the screen, I've got a brain. This is a little different than our brain. This is a monkey brain, but it's got sort of homologs to ours. So there's a big frontal cortex at the front. They have a parietal lobe, a temporal, an occipital. Same basic design as ours, just a little fewer creases and things, right? A little less surface area. But in the bottom of the temporal lobe, so if this is the temporal lobe on the side of the brain there, the bottom, what we've got is the inferior, that just means bottom, the lower part of the temporal lobe. So the inferior temporal cortex. There are neurons within the inferior temporal, the bottom of the temporal, in these monkeys that fire specifically when the monkey sees a monkey hand with fingers that are extended. So you could say it's sort of hand detector neurons, or even more specifically, neurons that only go off for open hands. So like what we see in 6 here, when we flash this in front of the screen, so you take a monkey, set them down in front of the screen just to test this stuff out and see how these neurons work. This is how we discover that they're specific to this thing. You show them something like 6, and this one neuron in the inferior temporal will go crazy. You'll hear the static sound when you show that in front of them. Show them a human-like hand, it'll still go off quite a bit. Show them a hand that's kind of closed, or right, it's sort of a shadow of what would be basically a hand shape, and they'll get some activity, a little less, but still quite a bit. Show them something kind of abstractly hand-like, and they'll get, a fair amount of activity, but less than. Show them some things like this that have a little bit in common with that hand, but not as much. You get quite a bit less activity, and then as you get further and further away, things that don't share as many features with an extended, finger monkey hand, then you get almost no activity. So those cells fire the least to these things. And so we can see that there are neurons further into the brain, right? We've left the visual cortex and the visual specific lobe, and now we're to the parts of the brain that combines information from visual senses and other senses. But we have neurons that specifically go off just if there's enough combos of visual input, the right combos of visual input to make something hand-like. So we could say those are hand neurons. But it's not just in monkeys. So we have the same kind of thing in the human brain, even when we move out of the occipital lobe and into places like the temporal lobe. So here you're looking at the picture of a brain from underneath. So back here, this weird looking texture, this is the cerebellum. And if you went up further, right, if you looked through the cerebellum, you would see the occipital lobe further up because we're looking from underneath the brain. But here we've got the temporal lobes along the side. Here's the frontal lobe up here. The temporal lobes along the side, we're looking from underneath. So we're looking at the bottom, the inferior part of the temporal lobe. And there's a little area in there, right? We have a copy on each side actually, but it's called the fusiform face area, because it's in the fusiform gyrus. Just happens to be where it's located. But we call it the fusiform face area, FFA, because it has neurons that only go off when we're looking at faces. So when you put faces in front of people, strangers' faces, faces they've seen before, there's neurons in there that will go off. You'll get a lot of activity in that area. Give them faces of animals, especially those that are close to humans. You get a lot of activity. Show them a sheep face or a face of a bird. Quite a bit less activity. Show them things that kind of have elements of faces, right? It's not actual faces, but things that kind of look a little bit like it, have some similar features. You get some activity there, but it's basically a face selective region with neurons that are selective to faces, which you could think of a face as really just a complex combination of simpler features. Things like lines and angles and combos of those things. A curve could really just be thought of as a straight line and then a slightly smaller angle, a straight line, then a slightly changed angle, slightly changed angle, slightly changed angle, slightly changed angle. So a bunch of V1 simple cells going off in a row, the correct combo of V1 simple cells is exactly what we would need to be able to recognize a curve like this that makes up part of lips or something like that. So really just to have neurons later in the brain, further into the brain, like in the temporal lobe along the sides there, we just have neurons that are wired up to a bunch of earlier neurons that only go off if the correct pattern of all that earlier stimulation goes off. But it allows us to get pretty complicated things, to be able to recognize complicated stuff. And we'll continue to see lots more examples of this. But I want to talk a little about neural encoding, how these receptive fields are used to encode information for things like specific faces. So, for example, there was a study showing the firing of an individual neuron in part of the temporal lobe that would go off when shown pics of Steve Carell, the actor, versus other famous people that were also used in the same study. So at the time they had Bill Clinton and I'm not sure, I forget who this is. Is that Drew Barrymore maybe? Anyway, other sort of well-known celebrity faces. And when they showed those control faces, so this little dotted line here is when they started showing the face in front of the person. Well, again, they're recording from an individual neuron in that person's fusiform face area. And when you turn on a picture of Bill Clinton, you don't see many neural spikes happening. Turn off the picture of Bill Clinton, right? Nothing much happened. Here they turned it on got a little bit of activity, but that could be totally random, right? Some noise. Here they turned on a picture of Drew Barrymore. You were getting a little activity even before they turned it on, but very little activity, not very much. So it's not like this neuron is saying, I'm seeing Steve Carell. But show various pictures of Steve Carell and you get, again, you turn it on or shortly after you turn it on, you start getting a crazy amount of activity. That neuron seems to be selective to Steve Carell. We could even say we seem to have found a Steve Carell neuron. If you're curious about this, by the way, you can Google, go to Wikipedia, look up grandmother neuron. There's this whole history behind this idea that an individual neuron might encode something like your grandmother, right? Being able to visually recognize your grandmother's face, or Steve Carell or Jennifer Aniston, or there's all sorts of other examples. It's not quite that simple, which is what I want to talk about for the last bit of this video. So the question then is, has experience trained our brain, right? You have to have, you're not born with a Steve Carell neuron. That doesn't make sense. You have to have seen a face over and over, had that experience for the wiring to happen through plasticity, right? But the question is, has that experience trained our brain to eventually hone an individual neuron that'll then fire specifically for each person we've ever seen? Not just the Steve Carells, but all the people we've met before, all the people with the specific faces. Is that how the brain works? If so, that's what we would call specificity coding. It would say the brain is so specific that it can get an individual neuron recognizing different people, like Bill, Mary, Raphael, whatever. So it'd be like cell 4. If cell 4 goes off, if neuron 4 in the fusiform face area, and again, there would be millions and millions and millions of neurons here, not just 10, but simplifying, zooming into this little chunk. Maybe there's an individual neuron that only fires when we look at Bill's face, but doesn't fire, right? Neuron 4 doesn't fire for Mary and doesn't ever fire for Raphael. It's very specific just to Bill. That would be specificity coding, right? Neuron 9 only goes off for Mary. It never goes off for anyone else, and so on. That's specificity coding. That is a little bit unlikely. It's not really supported by the data, at least not for visual encoding like this. Probably because there are just too many stimuli to have specific neurons for each one, like too many possible combinations of faces. Most neurons actually respond to a number of different stimuli. So most neurons, you pick out a neuron like 1 or 4 or whatever, but individual neurons, you'll see that they go off not just for one person, but for a variety of different people. And in fact, in the study, the Steve Carell study I showed you a moment ago, even the Steve Carell neuron fired for some other stimuli, like people or faces that are kind of more similar to him. Now, there's an alternative, a different way that information about different faces could be stored in a cluster of a bunch of neurons in a part of the brain like the fusiform face area. So an alternative would be not that we have individual neurons representing each person we've seen or each celebrity we're familiar with. Rather, population coding is about a whole population of neurons, a whole set of neurons. So it's saying a particular pattern of firing across a whole ton of neurons is what encodes a specific object or a specific person, a specific thing that we recognize. So thus, you could get way more combinations, way more stimuli being encoded because you can get so many different combinations. I mean, there's millions and millions of ways we could have these 10 neurons firing in different patterns, right? They're getting these bars all to the different sorts of combinations of heights. So even with just 10 neurons, we could get a huge combination of different faces recognized. But certainly if we have millions and millions of neurons in this, huge chunk of the brain, relatively speaking, in the fusiform face area, we're talking, multiple cubic centimeters. Like it's a large area. That's a buttload of neurons, a buttload of combinations, like an almost unimaginable number of combinations could be encoded if we use population coding, right? And so Bill might be represented as this specific pattern of firing across a whole bunch of neurons. And again, it might be thousands or more of neurons. That would be population coding. So that's an alternative. There's some evidence that it works kind of like that, but actually our best evidence is that the way things work is kind of somewhat in the middle here, kind of halfway between the two. So population coding is basically correct in that we don't have an individual neuron for each thing we recognize. It's not quite that simple. By the time we get up to stuff like faces and houses and complex stimuli like people we recognize, it's actually a bunch of neurons working together that represent that person, that fire when that person is in front of us. Population coding is basically correct, but it turns out it actually only requires a relatively small number of neurons to represent a distinct stimulus. So we use the term sparse coding as a sort of halfway between specificity and population coding. Or you could think of sparse coding as a toned down population coding, a very efficient population coding that uses the fewest neurons needed to be able to distinguish this stimulus from this stimulus from this stimulus. So just to kind of model this out in a pretend with just a simplified 10 neurons, we can see that across those 10 neurons, neuron 4 might have some activity for all 3 of these faces, right? And likewise, neuron 2 has different amounts of activity for these different faces here, but it's not selective to only Bill or only Raphael. And neuron 1 might be selective to only Raphael and not these 2, but maybe it also goes off for some other person's face. But we can have different combos of a smaller number of cells might be enough to distinguish things. That's sparse coding. That's basically how visual processing seems to work for most of the stuff we're interested in. Like once we get to complex things like recognizing objects and people. So this is kind of how I drew it out on a board. Like you could think of it like population coding is saying for, in blue here, my representation of Jennifer Aniston, recognizing that I'm looking at Jennifer Aniston in an old Friends episode, would be the activity of a bunch of neurons. I kind of drew out these blue lines really sloppily. Sorry for my bad handwriting and everything. But imagine like it's if all of those, maybe thousands plus, right? Thousands or more of neurons go off, that would represent Jennifer Aniston's face in a different combination, maybe involving overlap with a lot of the same neurons, but a different combo of thousands might make up my recognition of Bill Clinton or my grandma or whatever else, right? That's the idea of population coding. That totally could work, but it's almost unnecessarily complicated for the brain. But specificity coding, where I have an individual neuron that only goes off for Jennifer Aniston and one that goes off only for grandma and one that goes off only for Steve Carell, that doesn't seem to be how the brain works. It would require too many individual neurons because remember, each one, in order to have a Jennifer Aniston neuron, you have to have this neuron synapsed by or connected to, right, getting information from a bunch of other neurons that make up all the different combos of features that make up Jennifer Aniston's face and that make it slightly different from everyone else's face. That would just require so much complexity that we can actually simplify things in a sort of middle ground solution. The brain seems to use this sparse coding idea where I've drawn it out as a combo of just a few neurons. It's not literally just a few neurons that make up our representation of Bill Clinton or Jennifer Aniston or whatever that have that sort of combined receptive field of those. Rather, it's like a few percentage, a few percent of the neurons in that area rather than just a few of the neurons, but still much smaller than the full scale population coding would be. So we'll stop there for now. In the next video, we're going to move on to more complicated stuff, more high level stuff like recognizing people and things and using, information at a higher level than individual neurons.